Hi everyone, this is Mrs. Baker and I'm going to lead you through your notes on bacteria. Let's start with a question. What organelle does a bacterium lack that differs from a eukaryote? Make sure you have enough space to write questions on the side and space on the bottom for your summary. Bacteria are prokaryotes, which are unicellular organisms that lack a nucleus. That answers our first question. Typically, they range in size from 1 to 5 micrometers. And a micrometer is a millionth of a meter, so it's a very tiny, tiny little cell. Prokaryotes are identified. They are identified by characteristics such as shape, chemical nature of their cell walls, and the way they move and obtain energy. So let's start with shapes. There are 3 main shapes of bacteria. Draw 3 little circles so that you can draw examples of each. The first are bacilli, and I'm going to draw them. They kind of look like little rods or little sausages. Sometimes they link up like that. These are the rod-shaped bacilli. We also have cocci, which are circles. Sometimes they clump together, sometimes they make long chains. These are called cocci. They are spherical. And then the other typical kind we have are spirilla, which are spiral or corkscrew shaped. We can also identify bacteria based on the chemical composition of their cell walls. And there's a technique called gram staining that is used to identify different types of bacteria. So gram staining is a testing method that determines if bacteria's cell walls have a compound called peptidoglycan. What they basically do is they put chemicals on the cells, and then depending on what color they turn, it tells us whether they have this compound or not. And then we can put them into 2 groups, those that are gram positive and those that are gram negative. We also classify bacteria based on whether or not they have movement or what type of movement they have. Some bacteria are propelled or moved forward by flagella. And flagella are whip-like structures, or whip-like structures. They kind of look like little tails. So here's a little bacterium. That little tail behind it, that's the flagellum. Others move by secreting slime, so they slide around on the slime that they put out, or they may be carried by water or air. So they just get moved around through the elements. Bacteria can also obtain energy in different ways. Food and energy. Some bacteria are autotrophs, which means they make their own food. By using the sun, that's photosynthesis like plants do, or using chemicals. Some are chemotrophic. They take chemicals in their surrounding and make food out of them. While other bacteria are heterotrophs, and they need to take in nutrients by consuming other organisms or food made by other organisms. A lot of times when you're sick with a bacterial infection, you feel sick because the bacteria is cannibalizing your body. It's eating your cells or the products of your cells. It's using your cells for nutrition. Now, I'm kind of running out of room, so I'm going to bring in a note card to write about reproduction. There are three main forms of reproduction. The first is binary fission, which is a reproductive method or a reproductive process that involves only one parent cell. This is called asexual reproduction. And it results in two identical cells, similar to mitosis, where you start with one cell and you end up with two identical cells. The next form is called conjugation. And conjugation is when a hollow bridge is formed between two bacteria. And genes flow from one cell to the other. In a previous video, we showed you how we could have one bacteria with a little DNA in here, and another bacteria with another piece of DNA here, and they make this bridge, and a piece of the DNA literally flows from one to the other. So they're mixing up their DNA in this way. And then finally, endospore formation. And hopefully you're writing a little smaller than me because I'm going to run out of room and need another card. An endospore is a spore that is formed when a bacterium produces a thick internal wall. Oops, only one L. Internal wall that encloses its DNA. And I'll continue on another card that encloses its DNA, and it will remain dormant, kind of like in a state of hibernation, until more favorable growth conditions. So basically, if the cell, the bacterial cell, is in a place where it is not, there's not a lot of water, or there's not a lot of nutrients, it can go into hibernation and close up its DNA in this hard spore so that when things get better, it can come back to life and start reproducing again. So these are your notes on bacteria, and I'll see you next time.